Congratulations, parents, on the arrival of your new baby. With your consent, we took a sample of your baby's blood right after birth and ran it through the International Genomic Database. As a courtesy, we've compiled your child's disease risks, drug responses, and trait data. Please sit back while we inform you of your child's lifetime genomic profile. Sex, male. Length, 30 inches. Weight, 7 pounds, 8 ounces. We could have told you that. Your child is at a higher than average risk for developing type 2 diabetes. You need to limit your child's sugar intake. No soda or sweets, please. Make sure he exercises and don't let him get too overweight. Dad, you could stand to lose a few pounds yourself. Nearly 80% of people have the genotype that allows them to taste bitter foods, your child included. These people will generally not like the taste of Brussels sprouts. Recommendation: serve carrots. Parents seem to find this helpful. Your child seems to show an increased heart and lung capacity. Some people have called this the sprinting gene. We do not fully understand this yet, but it suggests your child might do well at sports like running or swimming. Recommendation: encourage track and field. It can't hurt, and maybe you can pay for college. Your child has tested positive for 1 copy of the Alzheimer's gene, increasing his chances of developing the disease from 10% to 30%. There is currently no treatment for Alzheimer's. We recommend you discourage boxing and football to avoid head injury, and perhaps you should purchase long-term care insurance early. And parents, you should be tested too. Your child is a carrier for the combination of genes commonly called the longevity genes. His projected lifespan is 93.4 years, barring an unforeseen tragic accident. This is the genomic manual that accompanies your child. Parents have been waiting for this manual for centuries. Please don't blow it. Good luck.
Well, good evening. Welcome to the program. The entertaining film you just saw, it begs the question, is this where we're going to be in the next 20 years? Every day when you open up the newspaper, you see about new discoveries, new genes being discovered for cancer, for heart disease, for diabetes. What does this mean to you in your health now? What is it going to mean to you in the next 5, 10, and 20 years? We're in the midst of a scientific revolution. This is made possible by the ability, the technology that allows us to map and sequence the human genome. And what will this mean to you? What will this mean in terms of what's called personalized medical care? The ability to not provide everyone with the same, but to tailor your care to the risk factors that you have inherent to you. To begin the evening, I want to make sure we're on a level playing field. And so when we're talking about a genome, we're talking about your genetic makeup, all of your DNA that goes up to make up who you are. One of our panelists, Francis Collins, talks about this being the instruction book for each of us, the instruction book for you. Now that this book is becoming possible, it raises quite a number of questions that we're going to be addressing tonight. Will we know at the time our child is born, like we saw in the film, everything about our child's risk, about their strengths, about their weaknesses? And if so, is this something that we do want to know? Do you want to know this when your child arrives, or is it something that would be frightening to you? Or would it be empowering? If you see in your own genome that you're at increased risk of heart disease? Would that say to you, I need to exercise and eat right? Or would it say that it's predetermined that I'm going to get heart disease? Why don't I go have a Big Mac? Those kind of issues are important to discuss. Are we emotionally prepared and educationally prepared for the kind of information that could be coming our way? There are lots of issues that deal with regulation. As a country, do we have protections in place so that if this information is available, it's in the hands of the right people? Who do you want to have access to your genetic information? Yourself, your doctor, your insurance company, your employer? How do we ensure that this information is kept safe? And what does this mean for the future of disease? Will we one day have the ability to select children who are born disease-free? And is that something that we would want to do as a society? Who decides what diseases should be eliminated and what diseases should not? And lastly, how predictive are our genes? Are our genes our destiny? Or are they less than that or more than that? Those questions are things that the folks here on the panel will be talking about tonight. So over the next 90 minutes, I want to explore with you, and we all want to explore with you, where we are today, how we got where we are today, which is a phenomenal story, what's possible with genomic testing, and as importantly, what's not possible right now with genomic testing, and what are some of the ethical challenges this presents. So essentially, at the end of the 90 minutes, hopefully you'll have information that you can use to evaluate what's coming down the pike. Because this is information that will be very important to your health and the health of your families. I don't think it gets any more personal than that. What we're talking about is about you. So to begin the evening, I want to introduce our very distinguished panel. It's an honor to be up here with these three individuals. First, on the end is Francis Collins. He's a physician geneticist, noted for his landmark discoveries of disease genes for cystic fibrosis and Huntington's disease, among others. He was at the helm of the Human Genome Project, which successfully mapped the first human reference genome. Currently, he's the director of the National Institutes of Health. He was appointed by President Obama in 2009. Sitting next to him is Robert Green. He's a professor of neurology, genetics, and epidemiology at Boston University Schools of Medicine and Public Health. He led the landmark REVEAL study to help better understand how genomics can be used in the early detection, treatment, and prevention of Alzheimer's disease. He's become so interested in translational genomics and personalized medicine, he's gone back to Harvard Medical School to do a fellowship in medical genetics. I think we could do a case study on that whole going back to school thing as well. And here on my right is George Church. He's the director of the Center for Computational Genetics at Harvard University, where his research focuses on next-generation genome sequencing technology. In 1984, his research pioneered early genome sequencing that helped set the stage for the Human Genome Project. Currently, he's the principal investigator of the Personal Genome Project, established to explore the legal, ethical, and social issues surrounding the age of personalized medicine. So the first part that we're going to talk about here is the current state of genomics. And Francis, I want to start with you on this one. The film we saw was rather tongue-in-cheek. But how far are we away from the kind of vision that was laid out there?
Well, we're pretty far away, I think, from any kind of societal consensus that this ought to be done in that kind of expansive way with every newborn, although I suspect we'll talk about that this evening. But we certainly have learned a prodigious amount in the last few years about genetic variations that play a role in risks for disease. Notice I said risks and not predetermination, and that's another topic we will come back to, I'm sure, on many occasions, because most of what we're reading in the DNA instruction book are not yes-no answers. They're things that increase your risk a little or decrease your risk a little. Think about this DNA instruction book as a book of information, a book that we're just beginning to learn how to read, written in a strange language that has just 4 letters in its alphabet, like you saw on the screen. They're actually chemical bases, but we abbreviate them A, C, G, and T. And there are 3 billion of them in one copy of the genome, and because you actually have 2 copies of the genome, one from your mom, one from your dad, you have 6 billion of those letters inside each cell of your body, and every time the cell has to divide, it has to copy the whole thing. That is a prodigious amount of information. If you decided to print out your genome just because it would be fun to have a hard copy, you'd need to be sure you had an appropriate storage facility, because if you did so on a hard copy with reasonable margins and reasonable font and reasonable thickness of paper and piled them up page after page, that would reach about the height of the Washington Monument in my town of Washington, D.C. And you have that information inside each cell of your body. And again, most of it you got from your parents, but actually you have maybe 60 to 100 mistakes in your genome that happened for the first time in you. Most of them probably aren't very important, but who knows. And then a whole bunch of others that you've inherited from your parents, which may or may not be important as far as your own health. Until we had that first copy of the human genome, and by the way, this is the 10th anniversary this month of the first draft version of the human DNA sequence, announced June 26, 2000, and then completed about 3 years later, because the last part is always the hardest. Until we had that reference, it was pretty hard to go in there and figure out exactly where are the variations that play a role in diabetes or heart disease or the common cancers, although we had begun to discover some of them. Now there's been this outpouring, hundreds of them in the last few years. But most of them are relatively weak in their contribution to disease. So is it time to find out for yourself? Now, it's pretty bright up here and it's pretty dark out there, but I want to take a little poll and then maybe we'll ask a different question later. So let me ask you, if at the end of this event, we would set up a little table out there in the hall and give you a little wooden spatula that you could scrape your cheek and get a few cells that have DNA in them, and we would promise to tell you next week what your human genome had to say about you, all 6 billion letters and some interpretation. Who would sign up? It's going to be free. Who would sign up? Okay. I think I see a lot of hands. I hope somebody who can see better is counting. Well, almost unanimous, I hear. Okay. So you all want to be empowered. You want this information. But is there benefit to this? This is a lot of what we're going to be talking about. What would you do with that information? You saw in the example, and something that Bob Green has worked on, Alzheimer's disease. We actually are pretty good at predicting who's at high risk and who's at low risk for Alzheimer's disease. But that one is one that a lot of people aren't sure they want to know. Some are, and Bob has done a very careful study to identify what happens when they get the information. But there's nothing at the moment that you can really do that we're sure of. We just had this big meeting at NIH to see if doing Sudoku would help you.
And the answer?
I'm really sorry to tell you. Doing Sudoku makes you better at Sudoku. And unfortunately, your Alzheimer's risk is unchanged. But if it makes you feel better, please go ahead. At least it makes people behave better on the subway. So, but there are other things we can learn from DNA testing that are quite valuable, quite actionable. We'll be talking about cancer, I'm sure, in a bit. And we're finding out you're at high risk for cancer. It gives you a chance to do something about it. Maybe not something that you would embrace, but it might save your life. What about diabetes? I'll get personal for a minute, because in the process of writing a book about all this, I decided I better go through this experience myself. So I had my DNA tested by one of these direct-to-consumer testing companies that will sell you analysis of your DNA. Not all 6 billion letters, but a carefully chosen 1 million that will tell you something about risks. And I actually had all 3 of the companies that do this test my DNA because I wanted to see if they'd get the same answer. And you know what? They did pretty well at getting the same answer as far as whether there was a T or a C at a particular position. But they interpreted it somewhat differently in some cases. And that's not because they're stupid. It's because the science is changing so fast. It depends on whether you use the paper that was published yesterday or just the ones that were published last week in terms of how you interpret the data. So anybody who gets on this particular train should realize these are early days. You're an early adopter. Things are going to change. The information you get will need to be rechecked next month, next year, 5 years from now, because your risk could potentially be reinterpreted in a different way.
Francis, I want to ask you a question. I read your book, Language of Life, and in there you talk about how the genome may actually play a role in what medicines are prescribed for you. Mm-hmm. Can you give an example of how that's relevant?
So when you get sick and a doctor prescribes a medicine that was actually the appropriate one for the appropriate diagnosis, I think we're all aware. It often works, but sometimes it doesn't. So what's that about? There are all kinds of reasons, but a lot of that is just variations in our own metabolism, and whether that drug was actually the right dose for you or whether its target in your body was actually there in the expected way. So this is a whole field called pharmacogenomics. And there are now, if you go and look at the labels on FDA-approved drugs, more than 10% of them say, before this drug is prescribed, some thought should be given to genetic differences between people. And there are a few where there's what's called a black box that says this drug really should not be prescribed without assessing that person's DNA because it may have a big effect on whether they're going to respond. Plavix, clopidogrel. Mm-hmm. Second most prescribed drug, I believe, in the country. About a third of people actually get no benefit from that drug because they lack enough of the enzyme that's necessary to convert the drug in the form that it's given to you into its active form. So they can take the pills, but the pills never get activated because of their genetics. When you want to know that before you spent your money and maybe ended up having that stroke or that heart attack that Plavix was supposed to prevent, but couldn't for you because of your genetics. So there's an actionable circumstance.
Wonderful.
For me, the other action was finding out my risk of diabetes. And you might say, well, okay, and we're all at risk for diabetes. Shouldn't you have done something to think about that ahead of time? Yeah, true. But finding out my risk was about 60% higher than the average person. Got my attention last summer, and if you had met with me last summer, you would have seen me about 25 pounds heavier than I am right now. It was a wake-up call. Now, that wake-up call should have been delivered anyway, but it was a teachable moment, something that made me think about my health behaviors and realize they weren't ideal.
We're going to come back to a number of those issues. George, you've been a leader on the technology side from the beginning. I was wondering if you could share with us where we were at the beginning, what type of tools were being used, and how we've gotten to where we are today.
Well, the beginning as goes way back, but in the, say, 70s, 80s, you could estimate that it would cost about $30 billion. We delivered, Francis just described, delivering a genome for $3 billion. And in a way, we're on kind of the same kind of curve that a computer industry is on that everybody knows about, where you get an improvement of 1.5-fold every year. And that multiplies and multiplies. And then more recently, it's gone from 1.5 per year to 10-fold per year. And this is unprecedented in any business that I know of, any enterprise where you get these factors of 10 every year for 5 years. And so now we're getting to the point where just today was announced that genomes are commercially available for as little as $10,000.
$10,000.
$10,000. This was announced just a few hours ago.
What's driven those changes? What type of advances have led to that kind of?
It's mainly the same sort of changes that resulted in large-scale integrated circuit progress. Is changing from putting things, parts together to microfabricating them and then shrinking. And so now all the DNA is the little dots on slides where you'll do billions of dots simultaneously, where before you might do 90 or so.
So I interviewed a family just last week who had all of their genomes sequenced for about $200,000.
Right.
So if they were doing that today,
40,000. Right.
Should have waited.
I wonder how they feel about that.
Yeah. Well, that family actually helped to create this technology. So I think early adopters have to be ready to do that sort of thing.
And when I've heard predictions that this may one day cost less than $1,000.
Well, in effect, it will be very close to free in the same sense that all sorts of sophisticated technology is available to the consumer for free, like Google Maps on your cell phone comes with your cell phone, basically, or your internet. And it's just a matter of who's going to reimburse it, who's going to make money on, you know, your insurance company can actually provide you with a service without ever discriminating against you, just by giving you the information that could help both you and them save money. And there are many other business models out there as well that could make it basically free. Once it's down in the $10,000 to $1,000 range, if you can't recover that kind of money in 80 years of you know, life, considering that the cost of being in a hospital for a day is comparable, then.
Well, you know, there are a number of companies that now will provide direct-to-consumer genetic testing. It's not your full genome, right. But Francis was saying he went through that. Yeah. Do these provide useful, actionable information to the general public?
They do, but it will be improved very quickly. So they were early entrants, and they wanted to secure a position. And what they do is they don't. If they find something that's highly actionable, then you can probably believe it. It's very accurate. On the other hand, a lot of the things they have, they're missing data. You know, if they say you're not at risk for breast cancer, you might want to get a second opinion. But if they say you are at risk, then you can start having a conversation with your physician about it.
Now, you're one of the pioneers who's had their own genome sequence. Is that right?
That's right.
Did you find information that was actionable for you?
So I think the way to, so I was asked by my institutional review board to be part of my own study, and which is fair, you know. There were a few things that we learned along the way, and we can talk about that in a later section. But I think the way, an interesting model right now, the way to think about it is it's not like your cell phone that you whip it out several times a day and look at your genome. I think it's much more like an insurance policy where, you know, you got fire insurance and accident insurance and dismemberment, and everybody gets this insurance, and nobody actually wants to cash in on it. Nobody wants to be dismembered. Right. Right? And so, yeah, my genome is very boring, and I'm very thankful for that.
And your genome you've made publicly available? Is that?
Yes, and the traits. Genomes without traits are not quite as easy to do research with. Yeah.
What do you mean with the traits?
Meaning you want to make all these connections, as Francis was alluding to, that help us get to the next round of sophistication to find things that are highly predictive and actionable, sometimes combinations of genes or combinations of genes with environments. And that requires that volunteers have to put their genes, something about their genes, their environments and traits out there, so that scientists all over the world can go in there and be geniuses and figure out how these things interconnect.
So Robert, you're the clinical frontline person. The information that we're hearing about, is this information that's currently useful to you as a clinical geneticist?
Well, one of the surprising things to me when I started learning about clinical genetics was that almost all genetics is risk genetics. You know, I had the idea that if you had a mutation, it was going to cause a disease and there was nothing you could do about it. And so I think I share that perception with a lot of the public, that it's very deterministic. And it turns out that it is not. Even in those cases where you've got a very dominant mutation that's running through a family, it can affect people at different points in their life, at different ages, and to different severities. So every gene is really a risk gene, and we're always talking about risk, and that was a big surprise to me. Now, I think what's happening is that there's a huge over-expectation of what genomics can tell us about our health. And that's gradually going to be corrected over time in society, partly because of education and research, and partly because people will go ahead and get their genomic information. And as I've heard from a lot of them, they'll say, what is this all? You know, is this all I've got? And they'll also have to understand a little bit more about statistics. We talked about that ahead of time, and they're going to have to understand that, wow, a 40% increased risk, that sounds pretty scary. But if your increase in risk goes from 1% to less than 2%, that really doesn't mean very much. And that's where we are with a lot of these things that are available today. Your risk of heart disease goes from 1.2% to 1.6%, And that's a significant percentage, but it's really still a very small risk. So I think that we've got to understand a little bit more about statistics, and we've got to sort of reality test or sanity test the genetic information that's coming at us.
And how are we going to find out which of the findings are truly predictive in terms of health versus something that is very interesting from a lab analysis?
Well, there are a lot of people looking at that very carefully. And what they're doing is they're taking these small changes and they're looking to see if it actually recategorizes risk. So for example, the Framingham study has a very careful categorization of risk. And if you're in a certain category, you ought to take certain drugs. And if you're in another category, you don't have to. So there's been an experiment, for example, where they took 101 cardiac risk SNPs. And you think, wow, 101 genes that give you information on risk for heart disease? And believe it or not, they couldn't recategorize anybody. Those SNPs, all the information in those genes didn't move anybody from one risk category to another. So if that's what's happening at the moment, it means that the genetic information really isn't very useful for taking to a doctor and changing their mind about how to treat you. And that's where we are with a lot of things at the moment. Now, I think we're going to be much farther along in the future. But I think that's part of the reason why this has been highly touted and in some cases disappointing.
I think everyone wants to get in on this. Yeah, George and Francis.
I mean, I think we have to balance. This is the news, but we have to balance that with the older story in clinical genetics, where there are about 1,800 gene tests that are considered highly predictive and highly actionable. Now, they're cast aside as being rare. They're individually rare, but they're collectively common. 10% of most populations are actually affected by this. Not carriers, but actually affected by them. And so there are things like phenylketonuria, which are 100% predictive in a certain sense, but they still illustrate Robert's point, because if you have PKU and you don't do anything about it, you're going to be mentally retarded. But if you have it and you do something about it, you make pretty radical changes in the diet to avoid an amino acid, then you have a 100% chance you're not going to be. So it's really 100% environmental at that point, but it's highly predictive and highly actionable. And I think it's very different from the cardiac story, which I think we also have to think about, but there are 1,800 of these.
So I want to jump in on that too, because I think George is right. There are diseases like cystic fibrosis or PKU or Huntington's disease where a single gene gone awry has a very strong predictive ability to say whether that disease is going to occur or not. We call those highly penetrant conditions, one of those genetics terms that only geneticists could love. But most diseases aren't like that. If you're talking about diabetes or the common cancers or heart disease, no single gene is going to be responsible for a high risk in those conditions. We already knew that because they don't follow patterns in families that Mendel would have approved of. They're much messier than that. For those conditions, you have many genes, no single one of which has a large effect. The other important point, though, in terms of Framingham is we have so far just scratched the surface in uncovering what the hereditary factors are for heart disease.
Absolutely.
Even though we've discovered 101, that's probably less than 10% of what's there. So as things get better, as we dig deeper into the genome, the predictive abilities are going to get better and better and better. And I think we know for many common diseases that your risk is heavily influenced by heredity. Family history is the strongest risk factor for heart disease. That means it's in there somewhere in the DNA. But we haven't identified it precisely yet. It's coming. Maybe another 3 to 5 years, Framingham will change their mind. But your point is well taken. Right now, if you get into this, it's a little bit more recreational than perhaps actionable in some instances. But it's going to change over time. And as I guess all of us are interested in seeing medical care shift maybe more towards prevention, this is not a bad way to start that effort.
Well, I think that it's going to be important for us to talk about as well how risk can be equated with disease.
In the early 1990s, 2 genes, BRCA1 and BRCA2, were identified. It turns out that if you're a woman and you have a misspelling in one of those genes, your risk of breast cancer lifelong is about 80%. And your risk of ovarian cancer is about 55%. So these are very serious risks indeed. We know from much research that's gone on in the last 15 years that there are things that women can do in that situation, but they are draconian and they're surgical. Many women in that situation now choose to have their ovaries removed after they complete childbearing because the ovarian cancer risk often does not occur until a little later in life. But then the hardest question is, what about prophylactic bilateral mastectomy to deal with the breast cancer risk? That does reduce the risk to a very low level, maybe just 1% from a little bit of residual tissue that's often left behind. But obviously at great personal and serious cost to one's body image and many other consequences for that person. There is reconstructive surgery that's now available that's a lot better than many people realize, but who would possibly volunteer to go through something of that sort if they didn't have to? The good news for families is that at least now people can find out whether they're at risk or not, and many women living in those families are now able to find out they didn't get the misspelled copy of the gene, and their risk of breast cancer is no higher than the average person. But still, it is a powerful, emotional kind of experience to live in this family and go through that kind of testing.
Well, you know, it's one thing for us to talk about that and the theoretical issues that are raised, but we want to share with you an excerpt of a film. This is called In the Family, and it appeared on PBS. It was done by a filmmaker named Joanna Rudnick, who herself carries the mutation in the BRCA gene. And she traveled around the country interviewing women who were going through this testing and going through this decision making. And in this clip, we're going to see the Hanke family at their genetic counseling session in Dr. Mary Claire King's lab. Just a warning, it is a very emotional clip that you're going to be seeing.
Sometimes after you get the results, your mind goes blank. Like after I heard the word cancer.
We're not finding out if we're getting cancer or not. We're finding
Well,
out.
I know that, but it's you know.
That's a little bit more dramatic.
News.
Julie Hanke carries the BRCA mutation, and her 3 daughters just tested for the mutation through Mary Claire King's lab. Tomorrow, they get their results.
Well, as you know, you were all included in this project because your mom, Julie, was found to carry this particular genetic mutation, BRCA1 gene. And we have offered testing through our lab for the 3 of you girls. And we have those results to give back today and to talk about. The results show that Stacey and Corinne, you do carry the genetic mutation. Rachel, you do not carry it. Okay. So that's what we have found.
We're going to spend the next hours explaining why this is going to be fine.
Right.
And that you are not going to have to go through what your mom has gone through.
Right.
Absolutely. We know it's fine. It's just,
Absolutely. Yeah.
Right. This is going to be fine.
Yeah. And there's been a lot of anticipation, I know. So what, I mean, obviously everybody's...
I've been doing this
upset.
34 years and I'm crying too.
I know. I know. It's always a lot. What are you, what's the main thought going on right now?
I feel fine. It's just hard when you have sisters because some people, like, I know she probably feels guilty that she doesn't have it. And that's why it's hard. Like, individually we take it, but then we know that others have guilt or you feel sad for other ones. So.
And isn't it amazing? And knowing, cognitively, one knows absolutely that it's completely chance. And it doesn't make you feel any less guilty.
Were you really expecting to be positive, Rachel, or sort of planning for it?
This is the opposite of what I want. Yeah.
It's okay.
I know.
Well, it's life-saving, and that's the only way that I look at it. And we knew going into this that we, you know, whoever had it or whoever didn't have it or whatever was going to impact our family. But Rachel, you cannot feel that way.
No.
You just can't. I'm sorry. I know you do, and your feelings are legitimate, but...
Just think of all the support you will have to be for your 2
Yes,
younger
that's
sisters.
right.
The burden on you is going to be substantial.
Yeah.
More than if you were mutation positive.
Well, no, she's worried because she doesn't have an excuse to get her ovaries.
I might have to have children.
They're off the hook, right? It's a little best, to be honest.
This will be okay for all of you.
Absolutely.
Right.
You now have control of the situation for the rest of your lives.
Right.
Very emotional clip, very tough decisions that these women are facing. And you know, how old are these women who are getting this information? And what kind of decisions are they needing to make about their reproductive future?
In the clip, certainly appears those are women in their 20s. There's been a debate about whether this kind of testing ought to even be offered to anyone younger than 18, because obviously the impact here is profound, especially at times where you're already pretty vulnerable about who you are, whether you're different. So generally this is not offered, even if the parents want it, for children younger than 18. And so they are now facing, and I'm sure the follow-up conversation we didn't get to see with Mary Claire was what the options are. And those are not things that will have to happen tomorrow for those 3 young women. It was interesting, wasn't it, that the girl who tested negative in many ways seemed to have the strongest reaction. This is survivor guilt. This is very analogous to what's described to people who survived the Holocaust and who, by a great good fortune, were able to stay alive, but somehow feel this burden of responsibility for those who did not. Well described in families of this sort, and those individuals need support too. And oftentimes it's assumed, oh, well, they'll be fine. It's the ones who got the bad news and who now have to face these medical decisions that need all the follow-up. Everybody needs follow-up in these families.
In watching that clip, I was struck by the supportive environment in which the information was given. And one of the issues or concerns that's raised is information that's coming direct to you in your mailbox. Mm-hmm. What kind of information might you one day get? Now, you're training to be a clinical geneticist, but what are your thoughts on how this information should be received and given, and what the capacities are in our country to do that?
Well, for example, some of the direct-to-consumer companies right now are including some of the more common BRCA1 mutations in their offering. And some of them are even offering this to children. So you could stop right there and ask a couple of questions. You could say, what in the world is the impact of a young person or a child getting this information outside the kind of medical and supportive context that we just saw there? But you can also say the other thing, because the direct-to-consumer company is only offering the most common mutation, someone could believe that they're getting fully tested. They could have a family history, they could order it online, they could misunderstand that because they don't have one mutation, they might still have many, many others. And therefore, they might be falsely reassured. And this is sort of the Scylla and Charybdis of this issue. On one hand, we don't want to scare people. We don't want to stimulate excessive medical costs, excessive medical surveillance, which could have iatrogenic harm. On the other hand, we don't want to falsely reassure people by telling them they're not going to get something that they might then go out and change their behavior. The classic example is if you tell somebody they have a gene that means they will be less likely to get cancer if they smoke, would that somehow reassure the smoker and somehow permit them to go on smoking or demotivate them?
Right.
So false reassurance and excessive fear and excessive testing are these dangers we have to navigate between.
Well, I think this is a good transition point to the questions around Alzheimer's disease, an area where you've done a lot of research, and the issues around what information people want to know, and what they do with that information. We were just talking about breast cancer, where it's something that's highly actionable, something where you can make a difference in terms of your long-term survival and getting cancer. Can you talk a little bit about the APOE4 gene, what that means to people, and what your experience was?
Sure. Well, James Watson, when he got his genome, said he wanted to learn everything about it except for one thing. There's a gene which gives you a feeling for your risk for Alzheimer's disease. If you have one copy of it, you're at about 3 times the risk of getting Alzheimer's disease. If you have 2 copies of it, you're at about 10 or 15 times the risk. So that's a substantial risk marker. And Alzheimer's disease is common, so it really means something in the world. So as a paradigm for this whole issue, we took the issue of disclosing ApoE for Alzheimer's disease to people and to ask the question, is it going to terrify them? Are they going to get a catastrophic reaction? And what are they going to do with the information? And just to sort of encapsulate 10 years of research and 3, now going on 4, randomized clinical trials into a couple of sentences.
You could be on television if you could do that.
Are you offering me a job, Richard?
Yeah, right.
Is that in the situation that we used, which was controlled, screening people for distress beforehand, counseling them after if they wanted, no one had a catastrophic reaction. Number 2, there were surprises. They changed some of their behavior. They took vitamins. They did more exercise. Number 3, they actually bought more long-term care insurance. This was the first study to show that people with a genetic marker would actually change their insurance purchasing habits. And number 4, there's a self-protection mechanism. So we just finished a paper called, I Know What You Told Me, but This Is What I Believe. And it turns out that even though they walked into our medical center and we said with all authority, well, this is your risk, people sort of said, you know what, I think I know what my risk is. And so there was almost an anchoring effect, and they didn't change that much. So we've discovered a lot of things about what it means to receive genetic results that I hope will become applicable from one gene really to all of them.
Francis, is the Alzheimer's situation something that we can learn lessons for in terms of other discoveries that are out there?
Oh, I think so. I think it is, and Bob's work has really laid out in the real world how this can be received in a way we can learn a lot from. As we talked about, as we get better and better at identifying the hereditary contributions to long lists of diseases so that they're as good at making predictions as we are now for Alzheimer's, that whole question is going to get raised. And if you're thinking about, would you want the test or not, it seems to me there are 3 things that you're going to want to know about that test. Is what kind of risk is it able to provide you with? Well, we talked about one, well, you know, here's a test that can tell you your risk is elevated by 40%, but if it was 1% to begin with, yeah. But if, on the other hand, it's Alzheimer's disease, which we know is common, and it might elevate your risk by 15-fold, well, that's a big one that you might want to pay attention to. So that's one factor. How much risk can this test actually provide you? Second, what's the burden of the disease? If it's cancer, well, you're probably pretty interested in knowing about that because we do have a sense that cancer is a bad disease and there are often things you can do about it if you're at high risk. But if it's, you know, wet earwax, you can test for that, by the way, not maybe quite as driven to get that information. And then the third factor is there an intervention? Alzheimer's? No. So maybe even though the risk is high and the burden is high, the intervention's not there. You might multiply those things together and still get, I don't want to go there. But if it's BRCA1, and the risk could be very high because you've got a strong family history, the burden is very high, and the intervention could be life-saving, then your interest goes up. I think while people probably don't do that little calculation when they're thinking about that, I think intuitively, that's probably what a lot of people do when they're trying to decide whether they want this information or not. And what we're going to be able to do as it gets better and better is to provide more accurate risks and more information about interventions that work. That's a lot of what I do as the director of the NIH, is to try to come up with ways that if you end up in one of these high-risk situations, we know what to tell you to do to reduce that risk.
Let's ask them how many people want. How many people out there would like to know if you have the ApoE4, which would increase your risk of Alzheimer's disease? So it looks like about half.
Yeah, how many would not want to know?
Yeah.
There were some abstainers, I think.
So which brings us to the question of whether really actionability is an appropriate measure. Because what we found was that people wanted to know for different reasons than simply stopping the disease. They wanted to know because they're information seekers. They wanted to know because they carried around anxiety and they thought that information could help them deal with it. They wanted to know for insurance. And they wanted to know because like Sergey Brin with Parkinson's disease, if you find out you have a risk for disease, you can become invested. You can become an advocate. You can follow the literature. You can follow. You can donate money. So they wanted to become involved. So there are a lot of reasons why people are interested in their risk besides medical actionability.
So that's a different kind of action, a different kind of intervention, a personal intervention as opposed to a medical one, and that should not be discounted. Certainly agree.
I want to come back to that insurance issue a little later. The purchase of long-term insurance after you have already seen your hand, which I think is pretty interesting.
I heard a few executives in the audience going, so that.
A few gasps there. But I want to shift to the area of what's it going to take to get us to the fulfilled potential of personalized medicine. I mean, you were saying at the beginning that it's been 10 years since the Human Genome Project was officially completed. At that time, President Clinton announced that this would lead to a new era of molecular medicine, an era that will bring new ways to prevent, diagnose, and treat disease. Some would say the progress has been really fast. Others would say it's been incredibly slow in terms of getting to the clinical situation, getting to the doctor's office, getting to the individual. So I wanted to focus on what your big ideas are. What's it going to take to get us from where we are right now to that fulfilled promise, that dream, the animation that we saw at the beginning. And to set this up, I want to first show a video. And I want to reflect first on the trip I took last week to California to interview this family of 4, the Wests, who had sequenced their genome. And they received 3 billion bits of data. And their daughter, who's 16, for her science project, is sitting there on her computer.
6 billion each.
Yes, each.
6 billion each, yeah.
6 billion, 3 billion pairs, 6 billion each. Yeah. Sitting on their computer with a spreadsheet trying to figure out what any of it means. So how do we get from there to what we're talking about here? So in this video, this is an excerpt from a documentary that's currently in production. It's called Genome, The Future Is Now. It's by filmmaker Marilyn Ness, who's also the producer of our evening tonight. And it's been following George Church and his experiment that's called the Personal Genome Project. Watch this.
In the summer of 2007, 10 people gave samples of their blood, saliva, skin cells, disease, and lifestyle information. They also agreed to have their genomes sequenced and posted on the internet for all the world to see.
Cardiac hypertrophy, is that the condition that often kills athletes in their prime?
Yes, yes.
So I have the rare version.
You do presumably have the rare version.
How our lives will unfold in public will foreshadow how the public views learning their own genomic information.
People are saying that the future of the genome is 20 years from now. I say it's happening now.
For the Personal Genome Project, what we're trying to do is figure out to what extent genes combined with environment impact almost every aspect of your body. Things like hair color, eye color, medical traits, and behavior. We're not only working on the technology, we're also trying to recruit human subjects. And people need to understand that it's much more identifying than any medical research has ever been. So the DNA will be informing about the deep medical issues, some of which we don't know yet. You can't remove and make anonymous their facial information or their DNA fingerprints, which are used in crime analysis, as everybody that's watched CSI knows. The Personal Genome Project is the broadest, most invasive study probably ever in medical genetics. I'm volunteer 1 at the request of the Harvard Institutional Review Board, who wanted me to get a dose of my own medicine and make sure that I would experience the full experience and see and maybe actively and rapidly respond to the threats and needs of the subjects. Hey, how you doing? Yeah. Hey, Joe, John, how are you? Yeah. So tomorrow we're going to have most of the 10 participants coming in and meeting with each other for the first time. I have to say thank you big time, because this is really asking more than probably anybody has asked before, except maybe the military. So thank you all for participating. I think the thing we worry about the most is that we're not worrying about the right things.
Fascinating. Now, your project is rather controversial. What's the mission and what's the controversy?
Well, the goal is very similar to many of the medical genetic research studies that Francis and others have been championing for many decades. The thing that's a little bit different here is enabling researchers all over the world and people who we don't know who's going to make the big breakthrough. It could be just about anybody. To get access to these complicated data. But the important thing is connecting full genome sequences with as much environmental and trait data as we can gather, and these will be moving targets as we go forward. But it's to make these connections between, among those three components.
Now, one of the areas of controversy is that you identify the subjects.
Right.
And that's something that you feel is very important. Why is that so important to really mining this information?
It's not so much we identify the subjects as we make sure the subjects understand that they can be identified. I mean, much more so than other, so a typical study might say, we will try our best to keep your identity a secret, and they'll give you a 10-page consent form, which many people don't read before they sign it. We actually require that they get 100% on an exam that walks them through all the likely and fairly unlikely scenarios we think might talk them out of it. Nevertheless, 16,000 volunteers have come forward so far. So
16,000?
16,000, yeah.
And how many have you sequenced so far?
So we've been waiting for the cost to, we've been working on getting the cost to come down. So we've done, we have 12 individuals who have either partial or complete genomes. About 5 of those are what would be called a complete genome, meaning about over 93% of the genome is done.
Now, the individuals who participate in this, the data is open to the research community. If they find...
It's open to the entire community.
The entire community,
yeah, right.
Research or not.
Well, we don't know how to judge somebody's a researcher. Somebody in India with a $12 computer might be the genius that makes the breakthrough.
Are there systems in place so that if something's found in someone's genome, the information gets back to them? Is that part of the deal in participating?
Well, I think this is a huge opportunity for participatory science. I mean, it's like astronomy, where you go in your backyard, except here you really are the expert on this subject. You know more about your body and your family's idiosyncrasies than any scientist does, at least before you give them the full medical history and ancestry. So it's a tremendous chance for participation, and people do in many studies say they want to see if they're going to participate in the study, they want to see what came out of it, not just generally, but about themselves. And I think that's, in a way, it's a responsibility.
Was there any requirement for family members to consent to the participation?
So the Institutional Review Board, reflecting many decades of decisions of this sort, at least in the United States, says the individual is the prime consideration. And so, no, they did not have to contact their family members. On the other hand, for our project, we felt that it was the best thing. And so we asked them to make sure that at least first-degree relatives, their immediate family, was enthusiastic about it. We have so many volunteers, we really want the most enthusiastic families. And to some extent, we're recruiting entire families where everybody's getting sequenced above a certain age.
And just so everyone's clear, why is it relevant that first-degree relatives?
Well, so to some extent, getting your genome doesn't tell you that much about yourself. But the little that it does tell you, you get 50% of it going to say your children, who might be too young to make the decision for themselves. And so it doesn't It only tells you 50%, but it could be, in principle, could be informative.
But now, see, part of what's controversial, and George has been sort of visionary in terms of this concept of participatory science, which is really brand new. It's making people collaborators in the research, which is incredible. But part of the controversy, isn't it, is that in this huge stack of the genome that you described, there are typographical errors, there are places where letters are missing, and there are letters switched around in many, many ways other than the known mutations like BRCA1 and BRCA2. So part of the question is, you find all these typos, you find all these rearrangements, what do you say to the individual there? And so people have talked about, yes, the $1,000 genome, or maybe even the $100 genome, and the $1 million interpretation, because it's going to be a huge bioinformatics challenge to figure out which associations are real, which you can believe, and which are meaningful. And there's that same balance again, scaring people or overly reassuring them.
So, in the video, there was the individual with the cardiomyopathy.
So that was Steven Pinker, who some of you may have read his books. He was being counseled by Joe Flicuria, who's in the same program that Robert is now in at Harvard Medical School. And what we felt we had to provide was some kind of software that represented the best interpretation, obviously in consultation with a very skilled clinical geneticist, but coalesced all the literature and the databases as best we could. And then we could, as a community, as a big community internationally, improve that software. It's not just a database, it's actually delivering a prioritized list where we say below this line, we don't think it's actionable or predictable. It's not clinically significant. You can look below the line. We're providing you the entire genome and even an interpretation of the entire genome, but we think this is the line, and that's subject to discussion. So in Steven Pinker's case, that was very preliminary data. It could have been wrong. It could have been a mistake in the sequencing. It turned out it was correct. We confirmed it in a CLIA lab, meaning a government certified lab with a different methodology. Then it could be wrong at the interpretation level.
Right.
Not because of the software, but because of the literature. And originally, that one was based on a very small number of cases and controls. And what we did was we pried loose a lot more cases and controls that were not in the literature. We went around and asked clinics and found a much larger set. And it got large enough that we decided that we had to tell, not in that scene, but a couple months later, we had decided that he should see a cardiologist to get a pretty non-invasive echocardiogram to see if the ventricular wall was thickening. And like he said, athletes do drop dead, and it's not part of your routine medical
And did he
care.
have the condition?
And he did not yet, but this is a late onset, can be a late onset disease. And so
Francis, you want to weigh in on this?
So yeah, this I think raises a really important issue. How are we going to get those answers? If we're talking about variations in the genome that are uncommon and whose indications for interventions are unknown, how are we ever going to know the answer? And it does seem the only way we will get that information is to have very large numbers of individuals who are willing to take part in research studies, where they are willing to reveal all of their medical experiences, where they're willing to actually have their environmental exposures carefully monitored, because that's a critical part of this whole gene-environment interaction that we haven't talked about, and who can then help us figure out what is a validated correlation. How do you know that that T in that position in that gene that Stephen had really has a certain risk, and how can you then help him deal with that? Because right now, for so much of this, we just don't know. And here's, George, why I think you've done a courageous thing by encouraging people to come forward and participate in research in a wide open way. But I don't know how many people, 16,000 is good, but how many people from certain groups will be interested in doing that who are a little suspicious about this. And so while that is a model, I think we also have to pursue models that do promise people as best we can, making it clear that there's no guarantees, that it is possible to be in those research studies and not have every high school student actually looking at your picture and laughing because of your crooked nose, which I'm sorry, maybe they are, but no. It's a serious concern for a lot of people that privacy matters. I mean, your study makes Mark Zuckerberg look like a privacy advocate. I mean, you guys are really doing a full Monty here.
How many?
Not everybody wants.
How many volunteers would you think would be enough to show in various groups that this actually is a model that's scalable? I agree we should have multiple models.
It's the
but
various
how many
groups
would you like
that
to see?
bothers me. Because one of the things I hope we learn from this is about health disparities. Why is it that certain diseases occur in some groups more often than others? Why do African American men get prostate cancer at a higher frequency than Caucasians or Asians? Why do Native Americans get diabetes? Those are traditionally groups who have been uneasy about research in general and particularly about genetic research. And there are reasons why they feel that way. And if you require for all of your studies that you have to do the full Monty, I can't see how you're going to end up with a representative sample. And I think in those situations, and many others who don't happen to come from traditionally underserved groups who are also anxious about this, they need another option. Being part of research ought to be something people see as a benefit and an opportunity. And if you make the bar so high that you've got to put your face on the internet and all of your medical records, you're depriving some people who would like to be part of this from the chance to do so.
Well, and also participatory research can only go so far. There are certain phenotypes. There's certain conditions which I don't think people can identify for themselves. How many people in the audience have headaches, but do you know if you have migraine headaches or cluster headaches or tension headaches or TMJ? How many people know they have a relative with cancer but don't know exactly what kind of cancer that they have? So, you know, you may have very, very informed participants, but there's always going to be a role for the epidemiologist who really focuses on phenotype as a way of enhancing that genotype-phenotype correlation.
Well, in the case of headaches, if it's in your medical records, that would be quite significant. If it says TMJ in your medical records, you don't need to self-report.
The reason we have to recruit over and over again in terms of phenotypes is because medical records are so bad in this country.
Robert, you have a couple proposals as to how to move the field forward. And
well,
one involves direct-to-consumer work.
Yeah, I'm fortunate that we just learned that we're likely to be funded by NIH for a I'm not even looking at him. I'm not even looking at him. And
this was not just decided, people. We have peer review.
He didn't even know about it. About the first ever study before and after direct-to-consumer testing, in which we're actually going to query people with the cooperation, this is very courageous, of the leading direct-to-consumer companies. Obviously, they're not representative of the population at large, but we think it's going to be a window into why people want genetic information and how they understand it and what they decide to do with it.
Will that give information in terms of what systems need to be in place to provide appropriate counseling? You've talked about the newborn screening model as a way that we might be able to go forward as a country with providing this information.
Well, I think it's going to provide some important clues, because if you find, for example, that someone hears they have a slightly increased risk of cancer, and then they rush out to their doctor and they get all sorts of x-rays and they get all sorts of invasive tests, we know that that person's driving up medical costs and actually driving up their risk to themselves out of proportion to what they're trying to prevent. So we really need to know what people are doing with the information, what they're doing with doctors. Now, newborn screening is a fascinating system because everybody in this room got a little heel prick when they were born, and that drop went to your state lab and they performed genetic tests on it. Who knew? And this is an amazing system because there are experts in the state lab who advise all the pediatricians around the state. So I'm just, brainstorming and have had the pleasure of continuing that with Francis here tonight about whether you could take the small number of geneticists in the country and use them as knowledge centers in that same way to help in a distributed manner all the primary care docs and all the regular docs somehow manage some of the pertinent information, the way that geneticists in state labs do with newborn screening. And so we're also looking at the idea for proposals to do that and see what happens when you actually try to integrate genomic information into a primary care practice.
As a general pediatrician, and I also used to train general pediatricians, I can tell you that most have absolutely no idea how to interpret this information or counsel patients appropriately.
It works, doesn't it? I mean, when you were a pediatrician...
On the newborn screening, it works. But the scale we're talking about here, the level of sophistication, are those who were trained for newborn screening. Do they have the sophistication to interpret what's coming down, or do we really need to undertake a level of training and develop a cadre of people that doesn't exist?
I think we do, and I think they may not necessarily be physicians. I'll tell you, physicians' assistants have gotten intensely interested in the personalized medicine revolution and see themselves as well positioned to play some of the role of interpretation and counseling. So do nurse practitioners. There are genetic counselors who do this professionally. You saw one in the film clip with Mary Claire King, but there's only about 2,000 of them in the entire country, and so the numbers are not going to be sufficient to handle the vast majority of individuals who want some feedback about their DNA results, but they could serve also as experts to handle the more complicated cases. But there's no question about it. The current situation where most physicians and physician extenders have limited or zero knowledge about genetics is not sustainable. But you know what's going to drive it? It's going to be patients who go and get their DNA analysis done over the internet and come in with their printout and say, "Doc, what does this mean?" And if you're the doc and you get that question twice in one day, you're going to start to say, okay, that's my threshold. It's time to learn this stuff. And you know what? The good news is it's not that complicated. This is not neurosurgery or, God help us, immunology, which I never understood. This is really straightforward stuff. Genetics is a series of simple principles and some understanding of statistics. And docs have to deal with statistics, maybe not very well, but you know, when you have your cholesterol measured and, well, you should do something because it reached a certain level, or your blood pressure, really what's going on there is a statistical risk based upon that number. That's a lot of what DNA testing is going to be like. And if we could handle cholesterol and we can handle blood pressure, we ought to be able to handle this too. If we just rev ourselves up and don't be scared of it and get the professionals empowered with usable information that they can quickly learn about and begin to apply.
George, before we came out here, we were talking earlier about how mathematics is taught in schools. And you had, I thought, a rather brilliant approach or suggestion for how it should be changed.
Well,
I don't know if you want to be
radical might be a better descriptor.
I think it is radical, but
the idea is, well, so I use mathematics in my profession, computational biology, but I would still say that almost everything that I learned in high school, for most people, algebra, trigonometry, and calculus, is far less relevant than statistics. Statistics is something that cuts across many fields. Social scientists use it, and policymakers use it. Everyone should use it. And it's really not as hard, in my opinion, as algebra, trigonometry, and calculus.
And very little is taught in schools. Yeah. Francis, I want to come back to you. Now, you are in a major policy position. So I don't know whether you want to comment on their big ideas, pose a big idea of your own, or punt. But I would give you the option.
A government employee punting? That's never happened.
I do have some specific questions if you don't want to hit the big idea. One of the areas of concern is around direct-to-consumer testing and the whole open field, and whether the governmental regulatory system is moving at the same speed as these advances. And I want to get your thoughts on that.
So that is a concern. Just before I do that, the big idea, I will take your bait there. Right. It does seem to me that if we really want to see personalized medicine emerge into the role that it could play and could in fact be quite beneficial in terms of reducing incidences of preventable disease by empowering people with their own information, by getting beyond one size fits all, we ought to really try to see what that takes. And one thing would be to get people more comfortable with using their own family medical history, which is available to you now by making some phone calls or talking to your relatives, and it's free. And yet it's not generally used effectively in prevention, and it could be. It's a genetic test, after all. Another thing would be to go ahead and get to the point where our electronic medical record system works, so that you had the ability to have information there when you need it. And as the cost of genetic sequencing comes down, to go ahead and get your genome placed into your medical record so it's there at the click of a mouse. And the place where that's going to matter a lot is this whole area of using genetic information to choose the right drug for the right person at the right time. Right now, we know a lot about that, but it's totally impractical for the doc who's about to write the prescription to get your DNA test, because it's like, well, where do I have to send the blood sample, and how long does it take to come back? If it's sitting there in your record and he just clicks and says, oh, you're the one that shouldn't have Plavix, let's give you this other drug, then you've got a system that actually can help people. But until you have both the electronic record and the genome sequence sitting there, you're inhibited from that practice. So we need all those things. And we need good policy. So now I'll come to your oversight question. There are, after all, if you go to the internet and punch in genetic test, there's some pretty weird stuff out there. I mean, there is. There are companies that will offer you testing about what kind of diet you should eat in order to lose 20 pounds in the next 2 weeks, and then they will sell you the nutritional supplements after they do your DNA test. And it is utterly scientifically bogus. There are even more troubling ones that will tell you whether your child is predisposed to ADHD and what you might want to do about that in terms of a nutritional supplement. Again, a DNA test based on fantasy, tests that will tell you whether your child should actually take this or that particular athletic training because of whether their genetics will make them succeed or not. Very, very unproven kinds of recommendations. And yet at the moment, the way the government oversees genetic testing is based on sort of a strange principle. If you're a laboratory and you make a kit that does a DNA test and you send that kit out to other laboratories, then the FDA is all over you, and they're going to make darn sure that that kit is valid and that it gives information that's trustworthy. But if you're a lab that does that test inside your own walls, and blood samples come to you, and you do the test, and then you send the results out, there's a little bit of checking to make sure you don't mix up samples, that your laboratory is sort of reasonably well organized. But there's nothing to find out whether that test actually gives useful information. These so-called home brews have kind of fallen underneath the radar. Well, there's now, I think, a lot of concern about whether that is a good system. That concern's actually been there for 10 or 12 years, repeatedly raised by various groups, including groups that report to the Secretary of HHS. The question is what to do about that, and the FDA is wrestling with that very question, because what you don't want to do is come in heavy-handed and shut down a new industry that could potentially be a big part of our future as far as real advances in personalized medicine. But you also want to do something to try to prevent bad outcomes. So far, I'm not sure there have been a lot of bad outcomes, but as the field grows bigger, there could be. So some kind of balanced approach to this is likely to be considered and maybe implemented in the not too distant future.
As consumers, folks in the audience who think that, well, after hearing this, they want to go out and get some testing done. How do they pick? How do they decide? How do they know that the results that are coming back to them are correct?
So I think if you go to the web and you look at what's being offered by one of these companies, you can pretty quickly get a sense about what are they citing as far as the scientific evidence. The credible companies will give you citations of publications in reputable journals, many of them, that you can actually click on, because we have made sure now that much of the medical literature is publicly accessible, and make sure that those publications actually exist. And you will see other examples of people who have gone through this who are real people. The ones that seem to be promising something that's too good to be true, well, it's probably not true.
George, should this information only be available through you and your doctor together rather than direct to consumer?
I think in this transition phase, it's terribly valuable to have the medical profession involved in collecting family history. I mean, you can do it, but then interpreting the family history and ordering tests. Now, there is also a role for educational materials, so the recreational genetics, like the Ancestry that the National Genographic Project does. Getting people to think about genetics, whatever it takes, is valuable, but you just need to know, the consumer needs to know what they're getting into. If they're getting into serious medicine, they should have their primary care physician at least involved in that, ideally a genetic counselor.
In the last part of our discussion here, I want to switch a little bit, talk about some of the ethical issues that are raised. The idea that with genetic testing, you can identify diseases also suggests that you're, through prenatal diagnosis or through selection, you'll be able to eliminate diseases. Who decides? Should a parent be allowed to go to a reproductive technology clinic and decide either now or in the future that they want an athletic boy who is free of the Alzheimer's gene and free of other genes? And who should decide that? Is that something that should be allowed? And this is a toss-up.
If it doesn't cause medical harm, it's going to be very hard to ethically prevent parents from intervening in their children. I mean, parents already provide their children every possible advantage. You know, an extra 20 points on their SAT. They will go to great extremes to provide that. You know, it's going to be ethically problematic both ways, to allow them to make these decisions which are idiosyncratic, or to prevent them from doing it.
And I think that it's important to realize that decisions like this are going on all the time right now. There's the story of the parents who have achondroplasia, who have dwarfism, and who selectively choose to terminate a pregnancy of a normal height child so that they can have a child who is like them. And so these are unusual situations, but in our peculiar medical care system, where you can sort of go to whatever office you want and convince somebody to do this for you, I think the danger arises where the pseudoscience that you referred to meets the choices about life and therapy. And that's the one area I'm really worried about, because I think that for the most part, reputable practitioners are attempting to do these things in very ethical ways. But there's this danger that with the whole society's move to disintermediation, to making everybody an expert on themselves and not needing the doctor, which is in some ways a very healthy move, that we're going to get to the point where we're making all these decisions without the facts. And that worries me a little.
And the facts are important here, because the kind of selection that you're talking about, at least at present, is only feasible by something called pre-implantation genetic diagnosis. So this is in vitro fertilization, where eggs from the mother and sperm from the father mix together, and you end up with maybe 8, 9, 10 embryos. It is possible then to take one cell off of each embryo after it's grown up to an 8-cell stage and do a DNA test and see what's there, and then decide which embryos to reimplant. And that was originally developed for couples who were at very high risk of having a child with Tay-Sachs disease, for instance, which is a horrible recessive disease. And they would have one chance in 4 that a child would have that, and no couple would choose to go through that horrible experience of a child who dies by about age 2 or 3 of a horrible neurologic and blinding condition. But PGD has become more and more accessible, and it's being used for more and more applications. In vitro fertilization clinics have traditionally been kind of left to do whatever the physicians involved felt was appropriate. But there's one sort of silver lining here in the scary scenarios about the designer babies. The idea that yuppie couples would come along with enough resources to go to an IVF clinic and say, okay, here, I want a boy, and I want him to be captain of the football team, and I want him to get A+ in math, and I want him to play first violin in the orchestra. So dial that in, would ya? If they were able to find a group that would do that for them, it would turn out very badly, because all of those things, intelligence, athletic ability, good looks, musical talent, are governed by a very long list of genes, no single one of which has more than a small effect. If you've got 8 to 10 embryos, you're not going to be able to optimize much of anything. And so that couple who's just paid the big bucks and figured they've won the lottery are going to ignore parenting, and they're going to have this kid who's at 16 up in his room smoking pot and listening to heavy metal music and flunking every course because the parents forgot about parenting. So it isn't going to work. So if somebody tries to sell you a designer baby, yeah, it's not a good plan.
What about the issue of children's rights and having information that a child might not want to know? It's kind of a corollary of your participants having to talk to their family, or something you suggested. What if it's a disease that manifests itself not in childhood but later, that doesn't have a prevention strategy in childhood? Should the child have the right to make the decision as to whether they find that out?
Well, at Children's Hospital in Boston, a team of people led by Ingrid Holm and Zach Kahani have created something called Gene Partnership, where they're specifically offering results back to parents of children that they hope to test in the future. And they've created a status where as the child gets to be about 7 years of age, they can learn certain types of information, and they're 14 years of age. With their parents' permission, they can learn most of it, and then when they're 18, they get all of it. So there is a precedent for sort of staging in a reasonable way, and with the parents' cooperation, the kind of information that you can give a child. I think that's a lot more appropriate approach than treating genetic information as if it were Facebook and allowing social networking on the basis of your genes.
Yeah, interesting. The issue of discrimination comes up, genetic discrimination. I mean, there's discrimination for all kinds of reasons. But the kind of information you can get from a genome is extremely detailed. The information that you have available to anyone on your subjects. What's in place now to prevent discrimination in the workplace, with health insurance, and is it enough to prevent more broad discrimination for genes?
So some of us worked hard on that issue over more than 10 years to try to get effective federal legislation in place to prevent genetic discrimination. And happily, 2 years ago, I had this chance to stand in the Oval Office as the president signed the Genetic Information Non-Discrimination Act. This will basically, and it does now, make illegal the use of your genetic information in health insurance decisions and in the workplace, which were the major concerns that people had. So that can no longer be done. That's a criminal act. But people should be aware that's not the only kind of discrimination that could be out there. It doesn't say anything about life insurance. It doesn't say anything about long-term care or disability. So those are still instances where your information could, if it falls into the hands of an insurance company, be used to set a particularly high premium if you're at high risk, or even exclude you altogether. And at the moment, there's no legal prohibition against that. And people should think about that before they learn about their own DNA in ways that they might later wish had remained out of sight.
Are there requirements currently to declare that information at the time you're buying insurance?
Well, I'm interested because George mentioned something, or maybe, I'm sorry, it was Bob mentioned something about your Alzheimer's study, who went out and actually bought more insurance. I don't know what the circumstances are in every policy, but most of the time when you apply for a policy, you say something to the effect that I have disclosed all relevant information. So how have they
But you made the suggestion, Francis, that they could buy the policy and honestly saying they don't know anything about it, and then go find out about it, and then drop the policy if they don't need it.
Well, we put it into our...
This is not a recommendation, folks.
You've heard it here.
Yeah. We put it into...
I didn't know you were going to bring that up.
Full disclosure, you know, right?
We put it into our consent form, Francis, that people had to realize the risk that if they learned information like their APOE, they might get asked about it when they were in the future buying policies. So some of them actually bought it before they entered the study. But according to what they said, what they told us, we had 5 times the number of people reporting that they purchased long-term care insurance if they were E4 positive than if they were E4 negative in the year after they learned their results. Now, when I talked about this in front of the International Meeting of Long-Term Care Underwriters in Cleveland, Ohio, Rio would have been nicer, but these little guys in bow ties and thick glasses, meek as they are, underwriters, virtually stormed the stage. They said, "You're going to put us out of business. You're going to take our business away." And I said, I'm not going to, because they were so incensed that the people could have inside information. And you do realize that a basic principle of insurance is that you can't have information beyond what the insurance company does, or you can game the system. So
yeah, it's called adverse selection.
Exactly. So it's a concern to the insurance company. I wouldn't be surprised if pretty soon long-term care insurance carriers start asking much more pointed questions.
They may require an Alzheimer's test, assuming that you will get it as soon as you get the policy. Right?
Right.
So, yeah. And they're exempt from GINA.
You know, I want to make sure that you don't walk away with a feeling that your genome is totally deterministic. And I want to ask you a question, Robert. James Watson, the co-discoverer of the double helix, he had his sequence revealed. You know what that showed?
Well, I'd love to hear Francis's take on it, but one thing it showed was that he was apparently homozygous for several rare diseases that cause mental retardation. Now, you got to think about that a second.
He could have been a contender.
That's right. If only he hadn't had those, think what he might have discovered. Well, they were almost surely technical errors, or they were, or there are things we simply don't know. Maybe it's possible. See, we don't come at it from that point of view. In the clinic, Richard, we've got a retarded individual in front of us, and we send off their genome. But we don't know if it works the other way around. We don't know if we take everybody's blood in the audience, we'll find 6 people who have genomes that would in the clinic, suggest retardation, but of course they're not because they're here.
I actually think that most of those have been sorted out now as either errors in the literature or errors in the sequence.
The prior probabilities of having the symptoms and having the family history create an entirely different set of circumstances when you're sending a genetic test. And we won't have that information unless we have a very large data set of population.
That's what we need. Well, we're going to need to wrap up this conversation. I think that it's going to be very interesting in 20 years to look back on the conversation and see what things we've talked about have come true, where we've moved with the field. Will babies be routinely screened when your baby comes to you in the delivery room or in your first visit? Will you have your child's full genomic lifetime profile? Will parents be informed? And will this data be protected in some way as different from some of the other types of health information that's out there? Or will you go see your doctor and you'll get an EKG because that's what they always do, and because nothing's been found that's a better predictor, and we really haven't moved along very far. I hope that after these 90 minutes you have a better understanding of this field and where it's going, and an understanding of the book that is you, and what may be coming that you're going to need to understand if you're going to take advantage of that. Before we close, we want to have a special closing. We have a surprise to end the evening, and the World Science Festival is falling between Mother's Day and Father's Day. So we have a special ode to all of the parents and children in the audience who've been living in pure genomic ignorance and bliss. So I want to welcome to the stage YouTube sensation, singer-songwriter, and almost Stanford College graduate, Adam Cole, who's going to perform for us a biologist's Mother's Day song.
Thank you. I've always dreamed of participating in a science conference at this level, and I never thought it would be in this form. But 1, 2, 3, 4. To make one me, you just add half of mom and half of dad. That is what I once believed. But now I know that I was wrong. You gave so much to me, Mom, besides 1/2 a set of genes. You gave me nutrients, transcription factors, nearly everything that matters, plus my prenatal environment, transplacental inheritance, mRNA mitochondria that back in the day once belonged to you, and I just want to thank you for supplying them. Just like 2 strands of DNA are spirally entwined, your nature and your nurture inspiringly combine. Scientists remind me, and I find that it is true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Mitochondria power the cell, and they have DNA as well. Transcription factors modulate transcription. And since they're in the cytoplasm, the egg's the only one who has them. And sperm, I guess they don't have the ambition. My sex determination gene means that I'm a guy. You gave me 1 X chromosome, Dad gave me 1 Y. X X has over 1,000 genes, Y has less than 92. That's why slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. I've roomed in your womb for 9 months, and I never once paid the rent. Your glucocorticoids help shape my hypothalamic development. I took your blood and sucked it dry of every nutrient. Sometimes I wonder where the time went. Sometimes I wonder where it all went. I know I'll never understand all you have done for me, but since you paid for college, I'll get my BS degree. And I have learned it's not BS, but absolutely true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Everybody. Nutrients, transcription factors, nearly everything that matters. mRNA, mitochondria, that back in the day once belonged to ya. Nutrients, transcription factors, nearly everything that matters. mRNA, mitochondria, that back in the day once belonged to ya. Singing nutrients, transcription factors, nearly everything that matters. mRNA, mitochondria, that back in the day once belonged to ya. Just like two strands of DNA are spirally entwined, your nature and your nurture inspiringly combined. Scientists remind me, and I find that it is true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Slightly more than half of me is thanks to you.
Well, I want to thank our panelists, Francis Collins, Robert Green, George Church. And I particularly want to thank Marilyn Ness, who put this all together and did a wonderful job. Have a good evening, everyone.